For centuries, the way we organized the natural world came down to an eye test. If an organism moved on land, swam in the water, or flew in the air, ancient philosophers like Aristotle grouped them together. We built our biological map based on physical form and behavior. As microscopes improved, taxonomists eventually split the microscopic world into a binary. On one side were eukaryotes, complex cells with a nucleus containing DNA. On the other were prokaryotes, simpler bacterial cells with no nucleus at all. But relying on physical traits creates a blind spot. Grouping organisms by what they look like obscures their actual evolutionary history, especially when dealing with microorganisms that look nearly identical under a lens. Until science found a way to read the internal genetic engine of a cell, our map of biology was missing the data necessary to see the true history of life on Earth. In 1977, an American biophysicist named Carl Woese proposed an alternative. We should stop classifying life by how it looks and start classifying it by the molecular machinery that powers it. His method centered on 16S ribosomal RNA, or rRNA, a component of ribosomes involved in protein synthesis. Because this molecule mutates at a slow and predictable rate, Woese used it as a molecular chronometer to trace evolutionary events back billions of years. By comparing these RNA sequences, Woese identified a group of methane-producing microorganisms, often surviving in environments like these boiling hot springs. Their genetic signatures revealed they weren't just unusual bacteria, but an entirely distinct ancient form of life. Woese named them archaea. This finding established a three-domain system, separating bacteria, archaea, and eukarya into three distinct evolutionary lineages. Adding a third branch to the tree of life made Woese a pivotal figure in biology. Yet his most debated hypothesis lies at the absolute base of that tree, waiting for modern genomics to validate it. Today, advanced genomic sequencing has uncovered an expansion of biodiversity known as candidate phylloradiation, or CPR bacteria. Researchers often refer to this vast, uncultivatable group as microbial dark matter. These unique organisms feature ultra-small, stripped-down genomes. They lack the genetic pathways required to synthesize their own amino acids or lipids. To survive, they must physically latch onto larger host bacteria. Because they cannot sustain themselves independently, CPR bacteria rely on horizontal gene transfer. Instead of passing DNA vertically from parent to offspring, they constantly steal and swap genetic material horizontally with their neighbors. These codependent biological networks serve as living modern echoes of the primordial environment Carl Woese predicted decades earlier. Woese's progenote hypothesis argues that at the oldest root of life, the first cell disappears. Early life was a communal pool sharing genetic innovations horizontally. This free-for-all stopped at the Darwinian threshold, crystallizing into isolated, vertically inherited lineages. The traditional tree of life only features distinct branches above this threshold. Below it, biology operated as a fluid, communal mesh. Modern science has now formalized Woese's hypothesis into a framework called Lucan, the latest universally common ancestral network. Under the Lucan model, the ancestor of all living things was not a lonely cell in the open ocean, but an interconnected protoplasmic network bound to mineral surfaces, sharing energy and genetic codes. The first autonomous cells did not simply evolve from this mesh. They escaped from it once they crossed the Darwinian threshold and established their own stable membranes. Viewing evolution strictly as a vertical progression is an incomplete picture. The foundation of biology requires horizontal integration and communal gene sharing. The rigid geometry of the traditional tree of life stands in contrast to the codependent reality we now observe in microbial dark matter. Carl Woese did more than identify archaea to add a third branch to our map. He provided the theoretical tools to rethink the physical structure of biology itself. As this composite illustrates, CPR bacteria prove Woese's physicalist view. Life is an interconnected, self-organizing thermodynamic system not just a collection of isolated species. Woese mapped the branches of life, but his true legacy was revealing that if you dig deep enough into our past, those branches disappear, leaving only a beautiful, unbroken web.